Yeah, I used to go into your lab and borrow your microwave to kill plankton. Anytime I wanted to kill plankton, I would do it. Okay, Andreas has worked in many areas of microbial ecology, and he's going to be talking to us now about the Guaymas Basin, where his hydrothermal work, but he has many other interests as well. So, students from 1:45 to 2:15, if you have a particular question, you just want to meet Andreas after the pizza, that would be fine for you. And after getting his PhD at MPI, Andreas went to Kuwait, where he was a postdoc first before we might be his last. Yeah, you were his last postdoc. And then was an assistant scientist there before he left to join one of my other good friends. So Andreas is very distinguished. He has well over 100 papers published in the field of microbial ecology, and he's also very modest. He's very read right now. And so, without further ado, let's hear about Guaymas.
Thank you for this wonderful introduction. Turn the speaker on so that you can all hear me. I was told there is the acoustics in the back of this room is really horrible. Can you all hear me when I speak like this? Okay. Today I'm telling you a little bit about my life with Guaymas Basin, deep sea hydro Gulf of California. It's a part of my life. There's a lot of other things. The other part of my life is in the Gulf of Mexico, working on hydrocarbon seeps and long-term ecological monitoring of the Deepwater Horizon oil spill. And there is yet another part of what I'm doing, focusing on deep subsurface sediments and deep subsurface microbiology. But when I was trying to think about what you would all like to hear, something that would be of interest to almost everybody, I hope, I thought, okay, it has to be Guaymas Basin. It's a spectacular hydrothermal vent site. It's not just a deep basalt-hosted vent site. It has lots of sediments. The sediments are organic rich. You see them here on, see, sediments, and a hydrothermal flow through them is sustaining massive microbial nets. So if you love microbial nets, microbial life in fullest abundance, then sooner or later in your life you have to go to Guaymas Basin. There is no way around it. Over time, since my postdoctoral advisor, Holger Janasch, introduced me to Guaymas Basin back in 1998, I have returned a couple of times. Most of what I'm telling you today are the fruits of my lab's united labor from cruises in 2008 and 2009. I had another short cruise last fall focusing on sediment coring, and we'll go again with Atlantis and Alvin, hopefully in spring of 2016. What I'm telling you today would just not be possible without the work of my many colleagues and collaborators at UNC, Dan Albert, Jennifer Biddle, Zina Cartman, Karen Lloyd, Barbara McGregor, Luke McKay, Howard Mendelwitz, and Brian White, just to mention a few of them, and collaborators from the Max Planck Institute for Marine Microbiology in Bremen, Germany, MARUM, the Center for Marine Environmental Research at Bremen University, and colleagues and friends at the University of Georgia. So, how do I turn the page? Guaymas Basin. It is the deepest spot in the center of the Gulf of California. It's one in a series of basins which actually form the northernmost extension of the East Pacific Rise as it splits Baja California and also extending northward as the San Andreas Fault, coastal California, away from the rest of North America. In a couple of million years, that slice of land will be an island separated by these ever-widening spreading centers. If you look into Guaymas Basin in detail, you see that the actual hydrothermal spreading center is going in a mostly north-south direction. The place that we are visiting is here in the southern trench. This is where most of the hydrothermal features are and where hydrothermal microbiology cruises like to return again and again. And the different spreading centers are then connected to each other by extensive transform faults, which then also continue northwards through California as the well-known San Andreas fault. Guaymas Basin works thanks to its massive sediment load and high sedimentary input of organic matter. It's a highly productive ocean region. Lots of organic material is reaching the sediment surface and gets buried over time. Imagine a several 100-meter thick sediment pack loaded with organic matter, and this is now slowly cooked by hydrothermal influence. You get, again and again, hydrothermal circulation going, carbon and energy sources are moving in, and all kinds of interesting microbial goodies, methane, sulfide, hydrogen, ammonia, DIC, and interestingly for Guaymas, lots of organic matter, organic, low molecular weight organic acids, and aromatic compounds. Petroleum, hydrothermal petroleum, is generated and is moving, migrating slowly back to the surface. Underway, we assume this mix already sustains microbial communities in the subsurface, and right at the surface, it is leading to these really flourishing, flamboyant microbial mat communities that we like to study. Tools of the trade, we mostly used beloved Alvin, the deep sea submersible that has been rebuilt completely last year, so now it is bigger, can go deeper, etc., but already back then, 2008 and 2009, it was our trusty workhorse. In spite of sometimes choppy weather conditions, I think only a single dive was missed, which is really important when you prepare a cruise for months and years and then run into bad weather. Fortunately, with Alvin, we could use almost the entire ship time for daily dives down to Guaymas Basin and up again. So I start with just giving you a general impression about how Guaymas Basin looks like. Here we are in the submersible with one of the pilots. You still see some light outside. After approximately 200 and 300 meters down, the light is fading, turning into darker and darker and darker blue. You will see bioluminescence of planktonic organisms, and finally that spectacle also fades. You are in the deep, pitch black deep sea. It takes 2 hours down to Guaymas Basin. You will be subjected to the musical selection of the pilot on the way. And at around 10 o'clock in the morning, you are down there and can look at the amazing surroundings. In the sediments, from time to time, you have massive hydrothermal mounds built up of hydrothermal precipitates, mixtures of carbonates and sulfides. If you could touch them, they would feel warm or sometimes hot to the touch. Gases seeping through them nourish all these microbial mats that are overgrowing them in luxurious patterns. And at the top of many mounds, you have sections where the channeled hydrothermal flow is reaching seawater. Here you see these Schlieren patterns, where hot hydrothermal fluid is emerging directly from the hydrothermal edifice. Around the orifice, you have new mineral formations growing at a rapid pace. This is actually the result of a recolonization experiment. After a chimney was taken away, after 15 days, a new one has been growing here. So hydrothermal growth of minerals and subsequent colonization of these minerals by microbes is a rapid affair in Guaymas Basin. You have hydrothermal megafauna, the classical poster child of hydrothermal vent microbiology and macrobiology. Riftia pachyptila, the tube worm, reaches the northernmost end of its range in Guaymas Basin. This is the perfect example of a chemosynthetic animal. It takes up sulfide, oxygen, and CO2 through its red gills, transports them into its interior body cavity, where not a normal digestive system is waiting, but instead specialized tissue full of bacteria, chemosynthetic bacteria, that are supplied by these energy-rich gases through the worm. And this is how the animal makes a living. It does not eat, it feeds its interior bacterial colony. These colonies depend, of course, on hydrothermal flow, and when hydrothermal flow ceases, then the days of rift year are numbered. Here we have a dying colony where scavengers are taking over. All kinds of little crabs and isopods. You see the beautiful, shiny red gills are gone, and the warm bodies decompose. So this efflorescence and death and rebirth of hydrothermal communities works on a very rapid pace, a couple of years. On top of this, we have the peak predator of Guaymas Basin. I had to include a picture. Benthic octopus species. We were actually chasing these beasts because one of our Mexican collaborators wanted them for lipid analysis and food web studies, isotopic food web studies. So with incredible luck, we finally caught 2 of them, but they are really wily. They can read the mind of the pilot. When the robot arm of Alvin is extended to grab them, they rise up, they do a little dance, and they dash away. Okay, but we are interested in microbes. So we keep our eyes on the sediments, where you have a constantly changing carpet of hydrothermal flow and associated microbial communities, here in a beautiful pattern arranged around a forming, just beginning mineral mound, flanked with a talus slope of beautiful microbial mats and sulfur precipitates. A little deep sea eel pound like this for scale. This is one of the most important tools of the trade, the heat flow temperature probe. You insert this thing into the sediment, all in all 40 centimeters. It has temperature sensors interspersed at 10 centimeter intervals. When you wait a couple of minutes, wait for the temperature reading. This is essential in order to determine sampling sites, because you want beautiful hydrothermal sediments, something really active. And of course, with a controlled temperature regimen, you want to know exactly the in situ temperature at different stages. After poking around with the help of the in situ temperature probe and exploring the terrain, we finally settled on the most beautiful microbial mats possible. Here we have the winner of the beauty contest, so-called marker 14. I explain why this is the winner of the beauty contest. The hydrothermal hotspots in Guaymas Basin have a distinct zonation. The hottest central parts are covered with these orangish Vegeatora mats. These are large filamentous sulfur oxidizers. It's its own special genotype. By measuring many mats, we found out that whenever you have the hottest spots, the orange Vegeatora will prefer them. So here we know this is the center of the hydrothermal flow. The marginal regions, where you still have some hydrothermal flow, are marked by, of course, yellowish sulfur precipitates. There's a lot of elemental sulfur in massive crusts in many places at the surface, and also whitish, slightly yellowish Vegeatora of a different type, which obviously survive on lesser degrees of hydrothermal flow, before finally you have the bare, brownish, olive-colored sediment. It does not mean that it is inactive. It just means that here the hydrothermal flow is insufficient to sustain a really flamboyant microbial mat. The mat winner, marker 14, was instrumented. Here we see in situ temperature loggers, which were measuring the temperature not only for 5 minutes, but over up to 8 days, constructed by Howard Mendlowitz, the ingenious chemical engineer, plus whatever else is required, in order to develop an understanding of the long-term thermal regime in these hydrothermal hotspots. The surrounding areas were, after 8 days, sampled for geochemistry and molecular biology and microbiology in order to finally get at the geochemical and microbial processes. In the first part of this talk, I will tell you a little bit about the bacteria that constitute the mats overlying these hydrothermal hotspots. Generally, they are described as members of the genus Vegeatora. I will leave out the taxonomic complexities. They sit on the surface and help themselves to hydrothermal reaction products which finally reach the sediment surface. Dissolved inorganic carbon, they have autotrophic capability, they like sulfide, there's a lot of sulfide in hydrothermal fluid here. They also like heterotrophic substrates. They have uptake systems, for example, for acetate. And acetate is produced by hydrothermal degradation of organic matter in the subsurface. In a way, these mat-forming bacteria are like the last gatekeeper that mediate biochemical exchange between the sediment and the water column. They intercept energy sources and also take electron acceptors, which they require for their metabolism, oxygen and in particular nitrate from the water column. So they are interface organisms that mediate chemical exchange between sediment and water column. We had the good fortune of having highly motivated collaborators from the Max Planck Institute for Marine Microbiology, which is a place like the Rockefeller Institute. They are swimming in money. They can build things for which you could buy the entire Department of Marine Sciences at North Carolina Chapel Hill, like this deep sea in situ profiler constructed by Dirk de Beers group. These things measure using microprofiler needles, tiny needles invisible here on this scale, which go in 25 micrometer increments into the sediment and then record things like pH, hydrogen sulfide, total sulfide, including other species, oxygen. Here we have different nitrogen oxides, N2O, redox status, and temperature. Everything in one machine. This turned out to be maybe the most precise way of actually getting at the environmental conditions right at the sediment-water interface where these microbial mats are living. It tells us surprising things, like although these are hot spots, the sediments get really hot, 100, 150 degrees, a few centimeters below the surface. The surface itself is always cool. Here in the temperature curve, you see 3 degrees, deep sea temperatures. So we have bacteria that, although they depend on hydrothermal flow, they would not survive without hydrothermal flow. They are not thermophiles. They are, in fact, psychrophiles. In order to keep them alive for a reasonable time in the lab, you have to put them into the cold room. And that for a hydrothermal organism. So highly counterintuitive. But this is the habitat preferences that they have developed over time. Here we have a slightly generalized sketch of the different physiological and geochemical roles. I already mentioned that Vegeator at the sediment surface intercepts sulfide and CO2 from hydrothermal sediment and uses oxygen and nitrate to oxidize the sulfide. The energy from this chemosynthetic reaction is used to assimilate CO2. In the sediment, you have different types of anaerobic organisms, sulfate respiring or methanogenic organisms, and consortia of sulfate-dependent methane-oxidizing archaea and bacteria, which mediate the oxidation of methane, a highly abundant carbon source, 10 to 20 millimolar in Guaymas vent fluid. Sulfate serves as the electron acceptor, sulfide and DIC are the outcome. The microbial degradation processes produce lots of sulfide and CO2 in addition to the hydrothermal load of the species. Everything reaches the sediment surface where Vegeator is waiting. The high temperature in the sediment, of course, now leads to enforces high temperature tolerance on all groups of subsurface bacteria, sulfate reducers, methanogens, methane-producing archaea, and we think also the anaerobic methane-oxidizing consortia. These things are originally all, without exception, described from cold seeps, cold temperatures, 3 to 5 degrees, maybe sometimes 10, but never ever more. Sulfate reducers, we know that they exist at temperatures up to 90 degrees Celsius. Methanogens, this is probably the biochemical group of organisms on the planet with the widest temperature range, from the freezing point to plus 110 degrees Celsius. And the high temperature representatives, guess where they come from? Guaymas Basin. Okay, first a closer look at the sulfur oxidizers. Here we have microscopic images under fluorescence. You see the two dominant species, the orange type and the larger white type on the peripheral sediments. Here you see a dark phase picture where the individual sulfur globules inside the filaments are lighting up. The organisms are taking up sulfide, oxidize it in a 2-electron transfer to elemental sulfur. They store the sulfur, and then they can oxidize it further in a 6-electron transfer to sulfate. The electron acceptor for this process, we are pretty sure, is nitrate. We will see why. These organisms have the enzymological equipment for nitrate respiration. Okay, first, step by step, we were spending time with habitat description. This is another take at the in situ temperature of these Beggiatora mats, measured with the relatively coarse tool of the in situ temperature probe. Even although this tool is much, it's not as sensitive as the in situ profiler, you can already see here that these organisms prefer a cool psychrophilic habitat. After dissecting the famous marker 14 map for geochemical profiles, sulfide, methane, DIC, temperatures, we came up with a scenario which was published, Deep Sea Research 1, where indeed in the hottest part of this mat you have the steepest hydrothermal profiles. The high temperature isotherms are squeezed towards the sediment surface. When you are moving away from this hot center towards the cold sediment, the gradients flatten out, the temperature gradient recedes until in the bare sediment there is not enough left. The different genotypes and physiological types of Beggiator, so is our working hypothesis, are dependent on these different hydrothermal regimes. For now we focused on the orange type, for which we had marker sequences, ribosomal RNA sequences, which placed these organisms as a separate lineage within the, meanwhile, very populous family Beggiatoaceae. Beggiatoaceae is a huge family of different sulfur oxidizers, many of them filamentous, many of them not, that were originally represented by just a single cultured species, Beggiator alba, from freshwater and nothing else. So this is an example where environmental surveys and molecular surveys have enormously expanded a range of the family. Okay, and we sequenced also the white organism, which turned out to be, again, a separate lineage. So the Guaymas Beggiatoaceae are their own thing. For the sake of convenience, we continue to call them Beggiator, but everybody knows that this is a catchphrase for taxonomically quite divergent organisms. Okay, one of the easy questions: what makes the orange Beggiator orange? Mineral precipitates? Is it some sort of biochemistry? Well, it turned out we harvested lots of this orange Beggiator mat, enough to extract proteins. The most abundant protein turned out to be an orange octaheme cytochrome. Here you see it isolated by polyacrylamide gel electrophoresis. So you didn't even need a sensational biochemical toolbox, just standard protein purification methods. Get the protein, send it out for sequencing, and then we had a draft genome of the orange Beggiator that was made possible by the Gordon and Betty Moore Foundation, and we found the gene for this protein embedded in the genome. Step 1. It's not a mineral precipitate. It's an enzyme synthesized by Beggiator. Question 2, what does it do? Gene homologies were sort of ambiguous. So in the end, Barbara McGregor, my colleague in Chapel Hill, who really took this on as a project closest to her heart. Without her, this would never have happened. Underwent all kinds of biochemical enzyme tests. And it turned out the functionality of this protein includes functions of anaerobic ammonia oxidation, the hydrazine oxidase, which you normally find only in Anamox bacteria, anaerobic ammonia oxidizers. It includes hydroxylamine oxidase, which is a key enzyme of aerobic ammonia oxidizers. And it includes nitrite reductase, which is an essential pathway of the nitrate respiration pathway. So all things considered, we think that the latter activity is the one that is physiologically important in this context. Since the organism has the genetic equipment for nitrate reduction, this activity makes sense. Vetiator does not have the genetic equipment for ammonia oxidation. So the other activities, which can clearly be measured, would not make sense in the genomic context as we see it right now. It doesn't really mean that the book is closed on these side activities, but it really means further research is required, and we are trying to do that now by cloning, expressing, and then testing the biochemical functions of additional genes and enzymes in the nitrate reduction pathway. These organisms living happily on the top of the volcano, oxidizing sulfide, of course, have relations to the microbial community underneath them. And here we were looking into unexplored territory. In particular, the problem of anaerobic methane oxidation. Since methane is one of the most abundant carbon sources in Guaymas Basin, it would be expected that the organisms in the sediment do something with it. Anaerobic methane oxidation has been described from many other environments. Andreas and her group have done pioneering work over 10 years to do a grand survey of the global biodiversity of anaerobic methane oxidizing archaea. In most cases, these turn out to be consortia. In red, we have the archaea. They are thought to be responsible for the methane oxidizing part, plucking the electrons from the methane. The electrons have to go somewhere. Sulfate is the electron acceptor. And this part is supposed to be done by the things in green, sulfate-reducing bacteria, which take over the electrons and use them to reduce sulfate to sulfide. It's an 8-electron transfer, turning methane into CO2 and sulfate into sulfide. The question is this possible at high temperatures in Guaymas Basin? So first, we looked at our database of methane isotopic signatures to look for clues. Over 100 individual measurement points were plotted in situ temperature versus delta C13 isotopic signature of methane. When you go into the high temperature range, 150 degrees and higher, this is certainly a range where microbial modification of the methane isotopic signature is not possible. So we decided this must be the range of the hydrothermal methane source, which fits quite closely with the reported literature values for Guaymas methane around -40. Now, biologically produced methane is lighter. Literature values go all the way from -50, -60, -70. Our sample set here is somewhat limited. We have some cool sediments where we think we hit on pure biogenic methane around -70. And then, of course, there are all kinds of mixtures of hydrothermal methane and biogenic methane, -40, where the biogenic signature is quickly drowned out by the overabundance of hydrothermal methane. Then, again and again, we have seen quite heavy methane, enriched in the heavy carbon isotope C13. And this happens when anaerobic methane oxidizers grab methane. They prefer the light methane, C12. So when they are done with their work, the leftover methane phase is enriched in C13. You see that as less negative isotopic values. So everything up here reveals the imprint of anaerobic methane oxidation. Question is, what is the temperature range? Well, if we assume naively that these data points reflect in situ processes, and if we forget about transport and mixing and other nasty factors, which certainly mess up the record, it looks like there's a boundary between 70 and 80 degrees Celsius. Below that, we can easily detect the isotopic imprint of anaerobic methane oxidation. At higher temperatures, it looks pretty much everything is hydrothermal. This turned out to be an unexpectedly good match to ex situ incubation experiments in the lab. Our collaborators from the Max Planck Institute, from Fritz Widdel's group, took Guaymas mud back home, incubated it at different temperatures, and then measured rates of anaerobic methane oxidation. And it turned out that the Guaymas mud samples are surprisingly good at anaerobic methane oxidation at temperatures up to 60, 70 degrees. 75 is maybe a little bit iffy. And also in the cultures, we have the requisite consortia growing according to plan. The stoichiometry also works sulfate and methane in a almost one-to-one stoichiometry as expected. So the process can be transplanted into the lab and runs at fairly high temperatures. Next thing, we wanted to identify the organisms that are responsible for it. Gene sequencing of different methane oxidizing sediments covered cool sediments, warm sediments, and really hot sediments. So what do we find in the really hot sediments? We find two branches. This is a tree of the universal marker gene 16S RNA, taxonomically and evolutionarily informative, which is used as a gold standard to identify microorganisms. We see one big group called ANMI1, anaerobic methanotrophic archaea, which is already known from cold seeps around the world. So they also occur here, and sometimes we find clones of these in hot spots, whether they really survive that or not. Then we found a separate evolutionary lineage, ANMI1 Guaymas, which has been found so far only in Guaymas Basin sediments and in a few diffuse venting locations on the East Pacific Rise, always associated with high temperatures. This is now our best candidate for organisms growing in hot hydrothermal sediments. We substantiated this result by parallel sequencing for a functional key gene of methanogenesis. Methanogenesis and methane oxidation use a highly conserved enzymatic apparatus with highly specific genes. The last one of which, methyl coenzyme M reductase alpha subunit, codes for the enzyme complex that catalyzes the conversion of cofactor-bound methyl groups into methane, and vice versa. It's a key enzyme of microbial methanogenesis and anaerobic methane oxidation. Do we find it again? As previously, we see the two dominant groups in hot locations, one of is congruent with ANMI1, and another one, exclusively Guaymas Basin, congruent with ANMI1 Guaymas. So we have two genetic signatures, 16S and MCRA, something to work with. In order to really improve on this scenario, we wanted to make sure that we get the habitat differentiation right. The previous data were obtained with samples which were collected in a haphazard fashion in different parts of Guaymas Basin. So by looking now at our prize-winning MET at marker 14, we thought, now we really look at the sediment in these three different temperature locations, slice it, and check whether the predicted habitat stratification for the ANMI archaea really holds up. Here we have bar diagrams of archaeal genetic signatures. This is a top layer, 13 degrees Celsius. This is a hotspot, orange mat, you will recognize it. Intermediate temperatures, the community is changing, and at bottom temperatures, 83 degrees, you will see that this black bar has taken over. What is a black bar? Well, this is the genetic signature for ANMI1 Guaymas. The organism obviously loves the high activity hotspot in the center of these mats. You do not see it outside of the center. This is a profile for the white Vegeatora mat, and this is a profile for outlying sediments. Its cousins also doing anaerobic methane oxidation in yellow, ANMI1, and some in brown. You see them also in other locations. But ANMI1 Guaymas is really limited to this hydrothermal hotspot. So this is now our best candidate for the toughest, most temperature-resistant group of methane-oxidizing microorganisms. We are superimposing now the results of multi-day temperature logging on these preliminary stratifications. The temperature profiles that we had used previously are just these 5-minute measurements associated with the hotspot, the white mat, and the bare sediment. Here you are now looking at temperature profiles over 8 days, color-coded. Screaming red is 100 degrees Celsius. Screaming pinkish purple is 0 degrees. You see how in the hot central portion the temperature quickly goes up, and when you are in the range around 10 centimeters, you have enormous hydrothermal fluctuation, also down at 15 centimeters. The temperature goes up and down all the time. So there seems to be hydrothermal pumping going on throughout the measurement period, meaning that the temperature fields in the sediments are not stationary, they are highly dynamic. This also means that the microorganisms inhabiting these sediments must be tolerant to fairly wide temperature ranges. Anything that lives here at 10 centimeters depth has to be prepared to deal with temperatures from sort of light blue, which would be 30, 35 degrees, to screaming green, lime green, which would be 60 to 65 degrees. So being a temperature specialist would just not work in Guaymas Basin. The organisms have to be adapted to a fairly wide temperature range. Now we superimpose the detection of these different anaerobic methanotroph lineages, and we see that the places where ANMI1 Guaymas occurs here is marked by strong fluctuations reaching the 65-degree boundary, and here deep down we detect it even at 80 to, who knows, a little bit higher, 90 degrees. Other ANMI groups, ANMI1, not ANMI1 Guaymas, the more commonly found ANMI1 is still found surprisingly in fairly hot sediments sometimes. ANMI1 has been described so far from cold seeps, but the version that we see in Guaymas Basin is still found here at 60 to 70 degrees. Also here, keeps company to some extent with ANMI1 Guaymas. When we come to really cool sediments, there's a different group that is taking over called ANMI2C, phylogenetically and probably physiologically quite distinct. These organisms survive with a little hydrothermal input, much meager assortment of methane, and not as reducing conditions. We should not forget that anaerobic methane oxidation is not the work of just the archaea. It's the work of a consortium. You remember the red and green microbial consortia. So we are also trying to keep an eye on the bacterial syntroph. This little tree is unfortunately distorted in scale, so I can tell you the bacterial target, it's a beast called hot seep group 1. It's officially uncultured. It's a separate phylogenetic lineage in the bacterial domain. You see it by fish hybridization associated with ANMI in hot environments, and by sequencing. You see it very consistently in hot sediments where you have lots of sulfide, where you have ANMI1 Guaymas, where you have microbial mats on top, indicating really active hydrothermal flux. So when these three conditions are set, lots of sulfide, lots of ANMI1 Guaymas, and microbial mat, then the organism comes up. So what we are working on right now is a collaborative endeavor with Brent Baker. He's a metagenomics expert. He originally worked with Jill Banfield. And after graduating from Michigan State University, he has now set up shop at the University of Texas, Port Aransas Marine Station, and starts to look into the metagenomic data set from Guaymas Basin sediments. That means all genes possible, everything that you can fish out, and then with high bioinformatics firepower, sorting these genes into piles, so-called genomic bins, which correspond more or less to individual groups of organisms, closely related organisms. And I told him already, ANMI1 Guaymas and the hot seep group 1 are high on the list of targets. So I gave him the samples which have, by other genetic screening techniques, the highest concentrations of these two target organisms. to make it a bit easier. Because if the metagenome is already dominated by genetic material from these two organisms, it should be easier to fish out and to reassemble. So they just got the second shipment from me a few days ago, and the PhD student, Kylie, says it's working well. They are working by assemblies. So I hope quite soon we get complete genomic assemblies. To review the overall evidence for high temperature methane oxidation in Guaymas Basin, we have evidence from the carbon isotopic signature of methane oxidation, which is visible in sediments with in situ temperatures up to 70 to 80 degrees. This happens to be broadly consistent with the UNMI range from in situ temperature logging, although the UNMI range based on that is a little bit higher, 80 degrees. The marker genes and functional key genes of anaerobic methane oxidation, in particular the high temperature Guaymas UNMI1 lineage, is found fairly consistently in hot sediments, again with, due to fluctuation, fairly high temperature range, 65 to 96. In the lab, this is probably a more conservative result. We see that 55 to 60 degrees is probably the best temperature for thermophilic anaerobic methane oxidation, and the temperature could go up to 70, 75 degrees, at least in the lab, before the process breaks down. So for now, the working hypothesis is a roughly 80-degree temperature window. The ultimate answer, of course, would require a pure culture, or something that is closest to a pure culture enrichment. So far, enrichment attempts have yielded the wrong UNMIs. So you get either nothing at all, because everything gets cooked, or when you try to go with high temperature, you get not UNMI1 Guaymas, you get only UNMI1, the previously studied sister lineage, which is also surprisingly thermotolerant. So we do not have a convincing strategy to get exactly UNMI1 Guaymas. There must be some environmental selection factor in nature that decides it. Maybe temperature, of course, but also something else. Again, we hope that the genome can give information. There are several important questions or complexes of ideas, roughly speaking, that emerge from this research into high temperature methane oxidation. Since methane is such an important carbon substrate, could these anaerobic methanotropes be the entrance portal for getting methane-derived carbon back into microbial biomass? It would be an alternative. Either the methane just bubbles out, or it seeps away, disappears in the ocean or in the atmosphere, or it goes into biomass. The evidence from stable carbon isotopic analysis of ribosomal RNA, scaffolding molecules of the ribosome, actually shows that it gets into the microbial community. Not just UNMI, archaea, but everything. In this complex plot, focus on this one. This is again work by Barbara McGregor, collaborating with Henrik Boschke in the Netherlands, who have developed the techniques for capturing these RNAs and subjecting them to isotopic analysis. Look at this middle line. Here is the background control. The isotopic signature of ribosomal RNA from superficial sediments without hydrothermal impact, around -15, -20. Here we have a couple of sites with hydrothermal impact. Here, admittedly, very moderate, and here stronger. And we get something around -30. So there is a difference. It is not as clear as we had hoped. Because privately, I had hoped to see isotopic signatures of -50 or something screaming at everybody in the environmental RNA signature. So in fact, it is more attenuated. But it really means the bacteria are getting carbon that is probably not photosynthetically derived, because this is what these guys have. They are helping themselves to some hydrothermal carbon, which could easily include methane. Of course, it can include hydrothermal DIC, and it can include hydrothermal acetate as well. We cannot really tell that apart at the moment. Going from this, at some point, I was running into two Guaymas aficionados in Woods Hole, Adam Sewell and Dan Lizaraldi, at Hui. And they introduced me to the idea that then you have this massive release of buried carbon that is mobilized as methane. Could it be that these methane assimilating organisms act as a kind of safety valve to prevent massive amount of methane escaping from the Guaymas Basin subsurface? Here we have this grand scheme of the Guaymas spreading center with axial venting. There's also off-axial venting. We know that now. The methane is released from the sediment package, and if it would go into the atmosphere, it would have consequences. Because, of course, you know that methane is a very powerful greenhouse gas on a molecule-by-molecule basis, 20 times as efficient as CO2. If it gets into the atmosphere in large amounts, it can certainly induce methane-derived global warming. And this is exactly what Dan and Adam had proposed previously in a Nature Geoscience article. They invoke massive outgassing of methane as a potential explanation for the thermal maximum at the Paleocene-Eocene boundary. And they regard Guaymas Basin as the modern-day analog for the trigger that caused this massive outburst of methane 55 million years ago. And in real life, that was supposed to be the opening of the North Atlantic. Well, the North Atlantic is open now, and the next big ocean that is opening is the Gulf of California. So Guaymas Basin has to be watched for this particular reason. So methane oxidizing archaea could be something like a brake on this geological process. Up to a degree, it could be that they catch methane, prevent it from entering the water column and the atmosphere, and counteract these outgassing and global warming events. Of course, only up to a point. When you have the whole Atlantic opening, maybe they really cannot do much. But it is an interesting idea to consider, and it is one of the driving factors behind further research plans in Guaymas Basin. We want to know how much of this methane assimilating microbial community actually extends into the deep sediments. And this requires deep drilling. Alvin push cores only go that much, 50 centimeters into the water column. With deep drilling, as proposed for the International Ocean Discovery Program, no longer drilling, it's discovery. Because drilling has a bad rep. We have submitted one back in 2014, got reviews back. The people liked the science, but they wanted site surveys. A site survey is important. We have to prove that there are no major hydrocarbon deposits which would blow up the ship. So we went back last October with a Mexican ship, the RV El Puma, and did gravity coring. Sampled all corners of Guaymas Basin. It's a fascinating environment. It's not just the hydrothermal spreading center. The off-axis bend sites and seep sites and nearby compaction-induced seep sites on the Sonora margin are there as well. So for anyone who loves sediments and the microbes living therein, it's a paradise. And the food is good. There will be a second site survey cruise next summer with the German ship Sonne, mostly focusing on 3D seismics in order to get a handle on the very complex topography in the sediments. It's a layer cake of basaltic lava sills and sediments, and another layer of sills, etc., etc. So drilling holes really have to be put with surgical precision into the spots where we want them to be. And in order to make more progress on the microbiology, and maybe we will have some testable hypothesis, then there is also an Alvin Atlantis cruise still scheduled for early 2016. So I have become something like a propaganda agent for Guaymas Basin, which leads me to write things like this. Frontiers in Microbiology, a very forgiving and tolerant journal. I happen to be one of the editors. So okay, so something like Biosphere Frontiers of Subsurface Life. It's not totally harebrained. This is actually based on data from DSDP Lag 64, where people measured hydrocarbons, and Doug Laroe from the University of Southern California did thermodynamic modeling and said, well, if you forget about the extreme temperatures and other obstacles, bacteria could actually get energy out of the sulfate-reducing oxidation of hydrothermally produced alkanes. So it's possible. And last but not least, before pizza, I would, of course, like to acknowledge some of my collaborators on these crazy cruises by showing their pictures. So if you run into them, Dan Albert has been responsible for much of the shipboard geochemistry, sulfate reduction measurements, completely tireless and always cheerful. Howard Mendelowitz, you already know about his role in designing the high temperature logger probes, plus lots of other things. So these two guys more or less carried Chris Martin's entire geochemistry laboratory away from Chapel Hill, reconstructed it on the ship, and shipped it back with nothing broken. My former PhD student, Karen Lloyd, now faculty at University of Tennessee, Knoxville, and my former postdoc, Jennifer Biddle, who got us started on the genome work with Beggiatoa by taking filaments and cleaning them so much that we could get finally clean genomes, which is a hard thing to do. Luke McKay, who did the work on marker MET14. Marker MET14 was his idea and brainchild. He selected the site, coordinated most of the research, and it's now a gigantic chapter in his PhD, which is almost transformed into a manuscript, but not quite. Barbara McGregor, who adopted the genomic analysis and proteomic analysis of the Beggiatoa mat. She's still continuing. She's getting currently paper number 6 out of the Beggiatoa genome. It turns out these beasts are so full of regulatory elements which you normally find only in filamentous bacteria. Filamentous sulfur oxidizers, filamentous cyanobacteria, and filamentous heterotrophs. In their evolutionary history, we must have traded all these regulatory triggers, so you find them now in their genomes. So the organism is much more interesting than we initially assumed. And here, Stephanie Meyer, one of the many Max Planck collaborators and postdocs who helped us in this endeavor. And I hope we have a little time for questions. Yes, please.
So I'm curious about the carbon that is recycled in the sediment pile.
Yeah.
And so obviously you're drawing microorganisms at the top of the pile in the region that you described, but their biomass is eventually buried and is part of the proportion of the carbon in the sediment. And that presumably becomes then the substrate that provides some of the recycled carbon that comes up and is utilized by the living organisms at the top. So I'm wondering in terms of carbon, the spectrum of carbon substrates. Well, first of all, I'm wondering what proportion of the total carbon in the sediment is in fact biomass at depth that's formed in that sediment as opposed to phytoplankton of biomass. And two, if they're fractionating against recycled methane that comes from that biomass, it's already going to be lighter, and maybe some of that so-called thermogenic methane isn't really thermogenic, it's recycled biomass, it's already light.
Yeah.
You see what I'm saying?
Your comment reflects the internal debate in the minds of many people who have looked at the methane isotope data. Jeff Seber, same. So the isotopic signature of the source methane around -40, it's in the gray territory between thermogenic and biogenic. And for this reason, when you look into the literature, people cannot quite make up their minds. Is it ultimately really thermogenic, like in hydrothermal vent sites which don't have this heterotrophic signature? Or could it be, to a large extent, biogenic? So it could be that the methane that reaches the sediment surface in super high fluids, 150 degrees, is already in fact a mixture. And again, one has to drill deeper into the source zone, maybe dozens of meters down, in order to see whether there are perhaps different methane fluxes in different settings. Maybe one type of methane flux may be originating from a sill, or something close to the spreading center, and maybe on the way, biological contributions are coming in to create the mix that you see at the surface. All that is possible. And in this view, then, the ultimately effective filter would be the filter at the sediment surface, where the carbon is ultimately caught by microbial mats and invertebrates. And microbial mats are, which I didn't show, infested with all kinds of grazing organisms. So there are little arthropods and everything in there, and they get eaten by something else, and ultimately everything leads to the octopus. So it's not exactly an answer. It's more an extended commentary to your commentary. But
At
yeah.
the top,
Yeah.
you measured the carbon pools at the top of the sediment pile.
Yeah.
What proportion of that carbon pool is now microbial biomass as opposed to an initial substrate
Okay.
from the water column?
Overall, the values are highly changeable. What has been done in the past between 3 and 12% organic matter, but that doesn't really distinguish between just organic matter and living microbial biomass. So that's an open question. Yes, please.
So the system that you're looking at, when there's no more methane or sulfide coming up, does all that microbial biomass go into the larger organisms, or how much of that is then released into the water column?
Water column release in Guaymas Basin you get mostly associated with hydrothermal plumes. And you have real orifices of chimneys.
Yeah.
My suspicion is that the sediment actually stays at the bentos because it is eaten by benthic organisms.
Yeah.
And when you go up the food chain, and even the octopus stays at the ground. So my initial working hypothesis is that what you have at the sediment surface stays close to the ground in Guaymas Basin, and only the carbon that is really injected by hydrothermal flow from chimneys, or the chemosynthetic substrates that are feeding bacteria in the clouds on the hydrothermal chimneys, that goes into the water column. Yes, go on.
So I was going to ask if you knew the dissolved oxygen concentrations right at the interface with Vegetor, but you say that there's lots of grazing sites, so it must be an oxygen environment.
It is, when you look at the microprofiler results, it goes down to 0 within millimeters. But the question is to actually define the interface. It's a little bit subjective. Then you would need a micro camera that tells you, okay, at this interval, this is exactly when the needle touches the uppermost layer of the mat, and now it goes through the mat. And now it really touches the sediment surface. Strictly speaking, you don't really see that based on the microprofiles. You sort of eyeball it. So what I would say, the invertebrates, they certainly have to be adapted to microoxic conditions, and maybe the occasional burst of sulfide when there is a blow of hydrothermal fluid coming out.
So my motivation
Yeah,
question
yeah.
is, why would Vegetor be nitrified? Why would they use nitrate rather than available concentration or no
Yeah. In theory, they should, but I think it is the fact that nitrate can be concentrated. These Vegetor have large intracellular vacuoles, like Thiomargarita, exactly. We measured nitrate goes up to 50 millimolar. So it's not exactly the same as in Thiomargarita, which goes up to 800 millimolar, but still. So, strictly speaking, they wouldn't need nitrate because they have some oxygen, but they have the nitrate accumulating vacuoles. And it's possible that they imported these nitrate accumulating vacuoles to the vent sites when they evolved to diversify and occupy this habitat. Or it could also be that sometimes they are really subjected to episodes of genuine anoxia. Oxygen is gone, and then they really need to survive on their nitrate, at least for some time. So which would mean, again, hydrothermal flow. It would also mean is they have to survive high temperature fluctuations. And this is a bit contradictory, because we tortured them in the lab. One of our Danish collaborators, Hans Roy, was doing that, and they don't like that. So 32 degrees is already borderline. more than a few minutes and they croak. So they are really living at the edge. They can take their breath, but they can't take the heat and so on. Yes, please.
Do you see a correlation between delta C13 and methane concentration for those enriched methane samples?
No. The concentrations are all over the map, and frankly, they are unreliable because they are always affected by outgassing.
So that's going to be tough. I mean, you should see one, because otherwise, to argue that you have a significant removal from methane, if you have a big fractionation but no change in concentration, tells me it's not important.
You see them in the sediment profiles. You can look at the profiles, and then you see that the methane is going down at the surface. But there is also a lot of wiggle curves in the methane profiles. And that is simply because it's overpressurized and there's outgassing. So the methane concentration is of iffy reliability, I would say. One has to really regard it with a question mark. One would have to recover pressurized sediment samples. So, and this is why I try to say, better don't interpret too much into that.
Yeah,
and I just want to see the methane isotopic signature should be less affected by just bubbling, so it should stay the same. So I regard this as a more reliable indicator.
In
Yes,
your
please.
micro, the profiler, where they had the max Planck profiler went down, there was a little oxygen maximum in the depth. I wonder if that was thought to be real or not.
Yeah, identified the bone of contention. There is a science paper, 92, Bobaker Jørgensen and his student, then-time student, Jens Gunnarsson, where they have similar profiles. So this is a prototype. They have little oxygen peaks underneath the mat. And that was interpreted as signs for hydrothermal pumping. It's obviously sucking little bubbles of seawater into the mat. So if you have also advective transport sideways, could be that sometimes you have that. Dirk de Beer, who has produced the new data, regards this as a fluke. The oxygen sensors could malfunction. We have seen some profiles which are so strange that they just cannot be true. There's no way. So he doesn't want to be so impolite and tell Bo that, no, your 92 oxygen profiles are rubbish. So we try to downplay this. So I say, okay, it could be hydrothermal fluctuation and bubbling, but since it's only one peak, too hard to tell.
But I would just say that it is a three-dimensional system.
It
So
is,
you
yeah.
don't, when you look at it as a vertical profile,
Yeah,
you
yeah.
are biased by the fact that you're
Yeah. You look only, yes, in one dimension. And of course, there is also advective flow going in different directions, depending on where you are exactly in the mat. So I hope that Dirk will recant and say, no, it's real, it's real. I have slept over it, and the profiler must work, it's real. Because it would fit beautifully with predictions. Yeah, I have to work on it. Dirk, don't you think it's real?
So now we'll think of pizza.
Yes, great.
Thank
Yes.
Andreas.
Yes.
I am officially moved into a new house. Really? Yes. Where is it? 5 minutes from my old place. Not even.